Hello, everyone, and welcome to today's webinar, Investigating the Conditionally Essential Role of Protein Ethylation in Proliferating Human Cells. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific Gibco. To learn more, please visit thermofisher.com/Gibco. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker, Guy Kunzmann, Graduate Research Assistant at UW-Madison. For a complete biography of today's speaker, click their name in the presenter's window at the top right of your screen. Guy, thanks for joining us. You may now begin your presentation.
Thank you for the introduction. Hello everyone, my name is Guy Kunzmann, and I am a PhD candidate at the University of Wisconsin. I'm very excited to give today's Cell Culture Heroes webinar on some of my ongoing thesis work investigating the conditionally essential role of protein ethylation in proliferating human cells. Just to introduce myself a little bit more, I grew up in North Carolina and attended North Carolina State University for my undergraduate degree. I got a bachelor's degree in biology in 2020, and during this time I really developed a passion for cell and molecular biology. This is perhaps not surprising, because outside of lab, one of my lifelong hobbies is baking bread, which sometimes just boils down to following a protocol and experimenting a little bit. After completing my bachelor's degree, I moved to the University of Wisconsin-Madison, where I'm currently pursuing my PhD in biochemistry. Today, I'm going to tell you a little bit about my ongoing thesis work, investigating how a cell's environment impacts its physiology. So there will be about 3 sections to my talk today. First, I will tell you a bit about the development of a physiologic culture medium, which I hope will be of interest to everyone in the crowd today. Then I will discuss some of the initial work done using this new culture medium, where my thesis advisor used forward genetic screens to investigate the impact of medium composition on gene essentiality. Finally, I will finish with my own thesis work, which is a follow-up to the genetic screens, focusing on a relatively understudied post-translational modification known as ethylation. As many in this audience are likely aware, in vitro cell culture is a near ubiquitous tool in mammalian cell biology and biomedical research. This is true whether the work is basic, aiming to define cellular pathways and biochemical mechanisms, or if the work is translational. For example, high-throughput drug screens on thousands of compounds across many doses are really only feasible in vitro. And so in biomedical research, in vitro models are often thought of as a starting point before moving into mouse or other animal models. However, we think of in vitro culture as a complementary system to mouse models, each with its own strengths and weaknesses. And cell culture has several distinct advantages as a model system. First, there's a broad scope of questions that may be asked, and these can be investigated in a variety of cell types derived from various tissues. Second, cell culture is very accessible compared to other model systems, and many of the necessary reagents are readily available to labs across the world. Third, in vitro culture is very flexible. You can create a wide variety of environments in which to culture your cells, and this includes changing things such as the culture medium of choice. And so to dive a bit deeper into this flexibility aspect, here I am showing several different environmental factors that can be controlled in cell culture experiments. Some of these we often take for granted, such as oxygen tension cells face, or the stiffness of the material that adherent cells are cultured on. But, as it turns out, many people have started to appreciate that conditions typically used for cell culture may not be optimal as a model of human physiology as we would like. To this end, there have been efforts to improve the modeling capacity of these in vitro culture systems, including controlling oxygen tension, varying the attachment substrate using 3D organoid models, and of course my favorite of these, changing the nutrient conditions cells see in culture, which is of course dictated by the culture medium of choice. Conventional culture media are typically formulated with varying concentrations of glucose, amino acid, vitamins, and salt ions. Here I'm showing a heat map of 4 conventional culture media and how their metabolite composition compares to human plasma. Blue boxes indicate a component is supplied at super physiologic concentrations, while white boxes indicate components that are absent from these basal media altogether. I hope you can appreciate that most of the heat map is either blue or white, indicating that these media do not reflect physiologic conditions. This is in part because these media were not created to model human physiology, but were rather designed over half a century ago to support cell growth and viability, as well as experimental consistency. However, human biofluids, such as plasma, contain thousands of metabolites, most at concentrations quite different from those in conventional media. So I hope you can appreciate that conventional culture media do not reflect physiologic metabolite concentrations. So, during his time as a postdoc, my advisor wanted to know if he could create a physiologic culture medium that more closely resembled the metabolic composition of human blood. He took a systematic approach to this, where he used a great resource, the Human Metabolome Database, to collect reported concentrations for over 2,000 endogenous metabolites in human biofluids. These were then passed through several filters, as each metabolite had to be a polar metabolite present in normal adult human plasma, and then, to maintain accessibility of this medium, each metabolite had to be above a certain concentration and available commercially. In sum, this led to a physiologic culture medium called human plasma-like medium, or HPLM, that contains over 70 total components, with over 30 of those uniquely defined in HPLM compared to conventional media. And so, in contrast to conventional media, which, as I mentioned, were designed to support cell growth and viability in vitro, HPLM is a physiologic culture medium designed to reflect the metabolite concentration of human plasma. Since the metabolite composition of HPLM is so different from conventional media, we next asked if culture in HPLM altered cellular metabolite abundances. And so here I'm showing a heat map of intracellular metabolites profiled by liquid chromatography mass spectrometry in 6 cell lines cultured in HPLM compared to RPMI. And I hope you all can appreciate the metabolite abundance has changed across many areas of metabolism, highlighting the impact of medium composition on cell physiology and metabolic processes. This was done during my advisor's time as a postdoc, and when he started his lab, he asked a question that may be deceptively simple. How do cells facilitate their growth? More specifically, he used CRISPR-based genetic screens to ask what genes are required for cell growth, and further, how does the environment impact which genes are required for cell growth? And of course, medium composition isn't the only environmental factor in cell culture, so I'm highlighting here that others have asked similar questions using CRISPR screens in varying oxygen tensions or in 2D culture versus 3D spheroid. However, our work focused on the impact of nutrient availability on gene essentiality, which is of course dictated by the culture medium of choice. And here is a schematic of our approach. We carried out CRISPR-based loss-of-function screens in the K562 chronic myelogenous leukemia cell line using a genome-wide sgRNA library targeting nearly all protein-coding genes in the human genome. These K562 cells were then transduced with lentivirus to introduce Cas9 and these sgRNAs into the cells, which were then split into either RPMI, the conventional medium of choice for blood cells, or HPLM, our physiologic culture medium. Analysis of these screen data identified hundreds of conditionally essential genes that collectively span many biological processes. Almost 3% of the targeted genes were identified as HPLM essential, or those genes that hurt cell growth worse when cells are grown in HPLM compared to RPMI. We also identified RPMI essential genes, which instead impact cell growth more when cells are grown in RPMI. Among those genes identified as RPMI essential were genes encoding for parts of the ethylation system, which is a ubiquitin-like post-translational modification that I'm showing in a simplified diagram to the right. It is important to note that all of the core components of the system were identified as RPMI essential in our screens, which is one reason why we chose to pursue this among the hundreds of conditionally essential genes. As I mentioned on the previous slide, ethylation is a ubiquitin-like post-translational modification that conjugates ubiquitin fold modifier 1, or UFM1, to target proteins. UFM1 is translated as an 85 residue protein that must be cleaved at its C terminus by UFM-specific proteases to generate its mature form. UFM is then conjugated to substrates through a ubiquitin-like 3-enzyme cascade to create an ethylate protein. In contrast to the ubiquitin system, there is only one of each of these enzymes, making this system very specific to UFM1. Ethmylation is also a dynamic PTM, and the UFM-specific proteases, or UFSPs, that act to cleave pro-UFM1 also participate in de-ethmylation of substrates. The primary ethmylated protein identified in cells is the 60S ribosomal subunit, RPL26. Several studies now have demonstrated that RPL26 ethmylation is crucial for ribosome quality control mechanisms to clear dysfunctional or damaged ribosomes, as well as selective autophagy of the ER, a process termed ER-phagy, where segments of the ER are targeted for degradation via ethmylation of RPL26. And while there are many great studies on this PTM so far, as a field, there's still much to learn about the biological role of this post-translational modification. To begin my work on ethmylation, I first wanted to validate the results of our genome-wide screens, that ethmylation is conditionally essential for cell growth in RPMI. To begin, I generated a clonal UFM1 knockout cell line using a single guide RNA targeting the UFM1 gene, and confirmed loss of UFM1 by Western blot compared to my control population of cells that instead express a guide RNA targeting an intergenic region of the AAVS1 locus. I then compared the growth of these knockout cells to my AAVS control in either RPMI or HPLM. Here you can see a greater relative growth defect when these cells are cultured in RPMI compared to HPLM. This differential growth defect is then normalized when I re-express a guide RNA resistant UFM1 cDNA, which confirms that this phenotype is on target and specific to loss of UFM1, which we consider validation of our pooled screen data. I next asked if the ethmylation system is conditionally expressed in one medium versus another, which could offer a simple explanation for this conditional essential phenotype. However, when measured by Western blot, we can see that each component of this system is expressed in K562 cells and does not vary with culture medium. Further, since RPL26 is the primary target of ethmylation, I next asked if medium composition impacted ethmylation of RPL26. Here, I'm showing a Western blot of UFM1 knockout cells expressing a flag-tagged UFM1 grown in either RPMI or HPLM. The primary band on this Western blot corresponds to ethmylated RPL26, which did not change in relative intensity between medium conditions, suggesting that ethmylation conditional essentiality may be independent of RPL26 ethmylation. I next asked which component was differentially available between RPMI and HPLM that could explain this conditional growth defect. As I mentioned in the intro, most culture media contain amino acids and salt ions, and HPLM has about 30 additional metabolites that are completely absent from conventional media formulations. So an easy place to start would be to add or subtract these components to RPMI or away from HPLM, and to measure the impact on cell growth. First, when I add these HPLM-specific components to RPMI, I do not see a boost in cell growth. And likewise, when I remove them from HPLM, there is again no change in cell growth, indicating that these HPLM-specific components are not the cause of the conditional growth defect in UFM1 knockout cells, and that this conditional growth defect is instead due to differential availability of an amino acid or salt ion between RPMI and HPLM. To address this, I next created HPLM derivative media with either amino acids, salt ions, or both adjusted to their RPMI-defined concentrations. I expect that one of these adjustments could induce this conditional growth defect in HPLM. When I adjust the amino acids in HPLM to their RPMI-defined concentrations, this did indeed induce this conditional growth defect. However, when I instead adjust the salt ions to their RPMI-defined concentrations, there is no change in growth between HPLM and this derivative media. Likewise, when I adjust both amino acids and salt ions to their RPMI-defined concentrations, I indeed induce this growth defect, suggesting that RPMI-defined amino acids are sufficient to induce a conditional growth defect in UFM1 knockout cells. I next asked which specific amino acid at its RPMI-defined concentration was responsible for this conditional growth defect in UFM1 knockout cells. To do this, I first divided the amino acids defined in RPMI into 4 subsets and then adjusted those subsets to their RPMI-defined levels, again in the context of HPLM. Here, I am again showing the conditional growth defect in RPMI versus HPLM, and then adjusting the amino acids in HPLM to their RPMI-defined concentrations, once again induced this conditional growth defect. Now I'm showing UFM1 knockout cell growth in HPLM with each of these 4 subsets adjusted to their RPMI-defined concentrations. As you can see here, only subset number 2 induced this growth defect, which contains about 7 amino acids. So I next asked which of these 7 amino acids could specifically induce this conditional growth defect at their RPMI-defined concentrations. And so I'm showing the concentrations in both RPMI and HPLM of these 7 amino acids. And once again, I am showing the conditional growth defect in RPMI compared to HPLM, and then inducing this growth defect when I adjust just this one subset of amino acids to their RPMI-defined concentrations. Now, among these 7 amino acids, when I adjust them to their RPMI-defined levels in HPLM, only alanine was able to induce this conditional growth defect at its RPMI-defined concentration. Alanine is actually absent from formulations of RPMI, despite being the second most abundant amino acid in human plasma. And so this is actually just removing alanine from HPLM is sufficient to induce this growth defect in UFM1 knockout cells. To confirm that physiologic alanine availability could support UFM1 knockout cell growth independent of medium condition, I next tested the growth of my UFM1 knockout cells in both RPMI and HPLM with or without physiologic 430 micromolar alanine. As you can see, alanine rescued growth independent of medium condition, and re-expression of UFM1 normalized growth across all of these conditions. So collectively, these experiments determined that ethylation is a conditionally essential PTM that depends on physiologic alanine availability. And so this raises the question of why UFM1 knockout cells depend on alanine, a non-essential amino acid, to support their growth. Alanine can be acquired by cells through import of the cell membrane, but in RPMI, alanine is not available. However, alanine can also be synthesized by the enzyme GPT2, which converts pyruvate and glutamate to alanine and alpha-ketoglutarate through a transamination reaction. Because of these 2 paths to alanine, it's generally considered a non-essential amino acid. But because GPT2 is the only path to synthesize alanine when it is not available outside of the cell, I considered if GPT2 activity may be impacted in some way by its ethylation, and if loss of ethylation could impact this biosynthetic path to alanine. However, ethylation is not a static protein modification, and endogenously ethylated proteins can sometimes be challenging to detect by Western blot. So here, I used a quick flag-based IP protocol by cloning a flag tag GPT2 and expressing it in K562 cells. I then enriched for GPT2 using flag-based affinity purification, and then immunoblotted this purified sample for UFM1. And so here is what this quick targeted approach looks like. So on the left, I am showing whole cell lysate of K562 cells expressing this flag tag GPT2 and blotting for UFM1. Here in the whole cell lysate, you can see that I did in fact pick up free UFM1, but I did not detect any substrates at the whole cell lysate level. However, when I enrich for GPT2, I also don't pick up any signal from UFM1 in this GPT2 enriched sample, independent of medium condition. This indicates that GPT2 is not ethylated, at least in these experimental conditions. And since I do not see that GPT2 is ethylated, I next considered if alanine catabolism instead was supporting UFM knockout cell growth. So rather than the biosynthesis of alanine through the transamination of pyruvate, instead catabolizing alanine to glutamate and pyruvate. And to determine if alanine catabolism was instead supporting UFM1 knockout cell growth, I used an isotopically labeled 15N alanine to trace patterns of nitrogen utilization by UFM1 knockout cells. Then, using LCMS-based isotope tracing, I measured fractional contribution of alanine-derived nitrogen to other amino acids, which can be generated by GPT-2 and subsequent transamination reactions to other amino acids, shown in the table at the right. Here, I'm showing unlabeled alanine in gray and the N+1 alanine label in blue, and their fractional abundance of alanine in the cell. And so we can see that when K562 cells are cultured in HPLM with this isotopically labeled alanine, it makes up about 1/3 of the total intracellular alanine pool, and this does not change in UFM1 knockout cells. This pattern does change when I measure the fractional contribution of alanine-derived nitrogen to glutamate, which makes up almost 10% of the glutamate in control cells, but almost none in UFM1 knockout cells. This pattern was consistent with the contribution of alanine-derived nitrogen to aspartate, as well as other amino acids that I have bolded. However, those not shown here all had contributions less than 4% even in control cells, so I don't consider this a substantial contribution to the total amino acid pool. However, collectively, these tracing data suggest a possible disruption in amino acid metabolism in UFM1 knockout cells, so I next measured the abundance of proteinogenic amino acids in UFM1 knockout cells compared to control cells using LCMS-based metabolite profiling. To start, I'm only showing the abundance of alanine in my UFM1 knockout cells compared to controls, and you can see when grown in RPMI, these cells do indeed have less alanine around compared to their growth in HPLM. Now, when I animate in the other 19 amino acids, I hope we can appreciate that most proteinogenic amino acids are depleted in UFM1 knockout cells, independent of medium condition. There are, of course, a few that actually increase in abundance compared to control cells, but the majority of these amino acids are about half as abundant in knockout cells compared to control cells, independent of medium condition. And when I re-express UFM1 in these knockout cells, which normalizes the growth of these cells in RPMI compared to HPLM, I hope you can appreciate that this also restores the amino acid abundance in these cells. So together, these metabolomic data show that amino acid homeostasis is disrupted in UFM1 knockout cells. And so now, just to take a step back, so far, I've shown that ethylation is a conditionally essential process for cell growth that depends on alanine availability, and that further, loss of ethylation alters both amino acid abundance and metabolism. And so as I haven't mentioned so far, I am only 3 years into my PhD, so this is very much still a work in progress. And I haven't completely unraveled the connection between protein ethylation and amino acid metabolism, but I am considering how a few stressors in my UFM1 knockout cells may activate central signaling pathways in the absence of ethylation. One event that is becoming well characterized in UFM1 knockout cells is an increase in ribosome stalling and a consequent increase in the endoplasmic reticulum stress response, or ER stress. Additionally, when UFM1 knockout cells are cultured in RPMI, they are limited by alanine availability, and we see a general depletion in their amino acid levels, which is a well-known activator of GCN2, which senses low amino acid levels in cells. There's also some crosstalk between these two stressors and their consequences, as ribosome stalling is another activator of GCN2. Further, both of these can converge on phosphorylation of the translation initiation factor EIF2 alpha and the transcription factor ATF4, which are known to impact transcriptional regulation of amino acid transporters and metabolic enzymes as part of the integrated stress response. And so I am still in the process of determining which of these signals, or if any, are activated in my system, and how alanine availability may impact these signaling pathways. But I have a sense that gene expression is likely altered in UFM1 knockout cells, and I do have experiments underway to determine if that is true. And so aside from the direct metabolic link to amino acid metabolism I just discussed, I also asked if alanine could complement for a specific part of the ethylation system. As I mentioned in the introduction, UFM is translated in its 85 residue pro form and must be cleaved at its C terminus by UFM-specific proteases before conjugation to substrates. In order to test if substrate conjugation is necessary for the conditionally essential role of UFM1, I also generated a truncated 82 residue variant lacking the C-terminal glycine required for conjugation to substrates. So here, once again, I am showing the medium-dependent growth defect of my knockout cells grown in RPMI, and the normalization of this growth defect when I re-express UFM1 in these knockout cells. However, when I re-express this truncated variant of UFM that cannot be conjugated to substrates, I see that this does not normalize the growth defect, indicating that conjugation of UFM1 to substrates is indeed necessary for its conditionally essential role. As an orthogonal experiment, I also knocked out UBA5, which is the first enzyme in the ufmylation system. Like UFM1 knockout, this should completely abolish ufmylation, and like UFM1 knockout cells, I found a conditional growth defect of UBA5 knockout cells in RPMI compared to HPLM, which was once again normalized by re-expression of UBA5 in these knockout cells. This provides good evidence that ufmylation of a specific substrate or substrates is required for its conditionally essential role in cell growth, but I next wanted to test how the dynamic aspect of this pathway supports cell growth. This time, I knocked out the more highly expressed deufmylating enzyme, UFSB2, which in addition to its role in cleaving the proform of UFM1 into its mature form, is also responsible for deufmylating proteins. And so once again, we can see that UFSB2 knockout cells harbor a conditional growth defect in RPMI compared to HPLM, and re-expression of UFSB2 cDNA normalizes this growth defect in RPMI compared to HPLM. And because UFSB2 is co-expressed with another deufmylase, UFSB1, I next asked if catalytic activity specific to UFSB2 is necessary for its conditionally essential role in cell growth. And so here, instead of RPMI compared to HPLM, I'm only showing the alanine-dependent growth defect of UFSB2 knockout cells in HPLM without or with alanine, and then re-expressing UFSB2 with a cDNA normalizes this growth defect in the alanine deplete conditions. I next re-expressed UFSB2, but a catalytically inactive variant, mutating the catalytic cysteine to a serine, and when I re-express this variant in my UFSB2 knockout cells, I see that this does not normalize the growth defect in alanine deplete conditions. Additionally, if I overexpress UFSP1 in these UFSP2 knockout cells, I once again see no change in knockout cell growth in HPLM lacking alanine. These data indicate that UFSP2 specific catalytic activity is required for the conditionally essential role of ufmylation in cell growth. And specifically, alanine can complement for loss of ufmylation and subsequent deufmylation of a substrate by UFSP2. Together, these data emphasize that ufmylation is a highly dynamic post-translational modification and is conditionally essential for cell growth, and it depends on these dynamics to fully support cell growth. Now this will necessitate the discovery of proteins whose ufmylation depends on alanine availability, which is another set of experiments that I have in progress but are not yet complete. In sum, today I have discussed the development of a physiologic culture medium designed to reflect the metabolic composition of human plasma. I then described the use of this medium to identify conditionally essential genes that depend on nutrient availability to support cell growth. And I then discussed one set of these conditionally essential genes, those involved in protein ufmylation, and how ufmylation impacts amino acid metabolism in proliferating cells. My biggest future direction will be proteomic experiments to identify differentially ufmylated proteins. And so with that, I want to thank everyone in my lab for their support during my PhD so far, of course my department and research institution for their support, my thesis committee for their guidance, and Thermo Fisher and Labroots for hosting this webinar. Lastly, I'd like to thank everyone here for attending, and I would be happy to take any questions. Thank you.
All right. Well, thank you so much, Guy, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. All right, we'll answer as many of your questions as we have time for. So let's get started. We've got some great questions already coming in. So, Guy, this first question asks, how do the metabolites in human plasma compare to model organisms such as mouse models?
Yeah, sure. That's a great question that we get often. So they have similarities and differences, and so a kind of key example would be uric acid, which is present at about 10 times higher concentrations in human plasma compared to mouse models, and can actually antagonize the activity of a classic chemotherapeutic 5-fluorouracil. And so this is sort of something we point to all the time as differences between mouse models and human plasma that actually have like real life impacts on the phenotypes we see. So there are differences and similarities. I would say alanine, specific to my story, is very similar between the human models and mouse models. So a mixed answer.
Great, thank you. Got another question here for you. Were any other ubiquitin-like modifiers identified in your genetic screens?
Sure. Yeah, that's another good question. Yes and no. So ufmylation was the only one we identified where the whole system popped out as hits in our screen, and that's one reason why we chose to study it, was because every single aspect strictly required for ufmylation was a hit in our screen. Some components weren't, such as UFSP1, and I suspect that's because it's very lowly expressed, and maybe UFSP2 could complement for that activity, but all of the core components hit. As far as other ubiquitin-like modifiers, we had some like E2s pop out if you dig into our data, or we had other PTMs, but not ubiquitin-like modifiers, and we identified as conditionally essential. So yes, there were others, but ufmylation was the one where the whole system came out.
Great, thank you, Guy. Now, why human plasma? Were any other biofluids considered when creating HPLM?
Sure, yeah. Human plasma, when we look at the human metabolome database, which is what my advisor used to create HPLM, there was just good quantitation and consistent quantitation of these components in human plasma, whereas other tissues might be a little bit more heterogeneous, and so maybe are different between individuals, different between different groups of folk. So human plasma was sort of the most accessible one to use, but there's certainly a merit to looking at other human biofluids as well and creating environments that mimic those.
Great, thank you. All right, next question here. From your amino acid metabolomics, I noticed serine abundance was actually increased in your knockout cells. What do you think is happening to cause that in contrast to other amino acids?
Sure, yeah. I'm not exactly sure on that one. My sort of running hypothesis is that you have like a disruption in amino acid homeostasis and levels for most amino acids. And yeah, serine is one that did pop out. Um, I have, so like I did for GPT-2, I did try and test if enzymes involved in biosynthesis of serine were ufmylated using the same method. And I did not identify any of the enzymes involved in that pathway as being modified by UFM1. Um, and so I'm not really sure what's going on there. Some other folks in our lab, when they profile their cells grown in HPLM, see a similar spike in serine. So it could be something specific to rewiring in media rather than a specific part of the ufmylation pathway. So that's just something I have to work through.
Great, thank you. Another question here. What happens to UFM1 KO cells later than 96 hours? And do the cells die out eventually or do they adapt?
Sure, yeah, that's a really interesting question that we haven't, I haven't necessarily looked into. We have a growth assay that we use that's about 96 hours to test sort of the short-term proliferative capacity of these cells. So I haven't necessarily looked longer than 96 hours. I would suspect that they would not necessarily adapt. I've done up to 96 hours. I've done growth curves, so I've checked points 12 hours apart within those 96 hours, and the growth defect tends to exacerbate with time. So I could expand that to say that that maybe that growth defect would get worse, but it is possible that they could adapt in culture as well. That's an interesting consideration.
Wonderful, great. Well, thank you so much, Guy. This will conclude our session for today. So before we go, I want to thank you, Guy Kunzmann, again for your time and your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific Gibco, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.